Auzu billahi minash shaitanir rajim. Bismillahir rahmanir rahim. Uh, today I am going to give a presentation on the topic of gene and genetic code, which will be submitted to the respected Dr. Mahmood-ul-Hassan. So first of all, we are going to talk about what is a gene. The term gene was first introduced by a Danish botanist in 1900 and another scientist or a geneticist called William Johnson in 1909. It is basically inspired by the Greek language, a word in the Greek language, gonos, which means offspring or procreation. So the term gene was first introduced by two scientists in 1909, and this term is inspired or derived from Greek language. We can define a gene as the basic physical and the functional unit of heredity, or we can also define gene as generation or a birth. A gene is the basic physical and functional unit of heredity. In biology, in biological science, a gene means generation or birth in a basic unit of heredity and a sequence of nucleotides in a DNA or RNA that encodes the synthesis of a gene product, either RNA or protein. Genes are made up of DNA. As you can see in this diagram, that this is a chromosome, and when you unwind the chromosome, this is the DNA after unwinding a chromosome, and this is all of this is DNA, and a specific portion of DNA here, as you can see, is labeled gene, which means that gene is made up of DNA, and it's along the entire length of DNA there are many genes. So gene is basically inside DNA, or you can also say that gene is made up of DNA. Some genes act as instructions to make molecules called proteins. However, many genes do not code for proteins, which means that throughout the entire length of DNA, genes are present, and most of these genes encode information which is required to make functional proteins and enzymes, but many other genes along the length of this DNA may not have any information, any encoded information needed to create proteins. So we can say that amongst all the genes present in a DNA, some genes encode for proteins and some genes do not encode for any proteins. Now we are going to talk about the alternative form of gene. In broad sense, the alternate form of a gene is called an allele. Every person has two copies of each gene, one inherited from each parent. Like, each parent contributes one alternative form of a gene, and that alternative form of a gene is called an allele. Alleles are forms of the same gene with small differences in their sequences of DNA bases. The short, in a short way, in a simple way, we can say that an allele is a variant or a variation or a different form or an alternative form of a gene. When the copies of a gene differ from each other, they are known as alleles. So this is basically a concept of allele, that what is an allele and how can we define an allele, and why allele is called an alternative form of a gene. As you can see in this slide, alleles may occur in pairs. Alleles may also be multiple, affecting the expression, the phenotype of a particular trait. The combination of alleles that an organism carries constitutes its genotype. If the paired alleles are the same, the organism's genotype is said to be homozygous for that trait, and if they are different, the organism's genotype is said to be heterozygous. As you can see in this diagram, in this picture, that on this chromosome we have allele 1, and on the other chromosome we have allele 2. So these two are different alleles. The gene is the same, like it's this light blue one is the same gene. This is the same gene, but it has two alternative forms on two different chromosomes. It's the same gene, but as you can see on this chromosome it has an alternative form named allele 1, and in this chromosome it has an alternative form named allele 2. So this is the concept of allele. The gene is one, like it's the same gene, but it has two alternative forms on two different chromosomes named allele 1 and allele 2. And just as I've explained earlier, the pairing of alleles, in this diagram you can see that when these alleles can pair in different forms. If allele 1 is paired with allele 1, it becomes homozygous. If this allele 2 is paired with allele 2, it also becomes homozygous. But if this variant form, this different form, like allele 1 is dominant and allele 2 is recessive, and if these two different or variant forms of alleles of one gene combine, then they constitute a heterozygous allele pairing. So this is the basic concept of alleles and allelic pairing, and how homozygous and heterozygous pairing is made. Now we are going to talk about the chemical structure of a gene. Genes are composed of deoxyribonucleic acid, as we all know, but except in some viruses where RNA makes up the genes, which means that genes are basically constituted of DNA, but in most life form, like humans, bacteria, and plants and fungi. But in some viruses, the genes are made up of ribonucleic acid instead of deoxyribonucleic acid. A DNA molecule is so as we have said that genes are made up of DNA, we now have to understand the chemical structure of DNA, because it constitutes genes. A DNA molecule is composed of two chains of nucleotides that wind about each other to resemble a twisted ladder, which means that the DNA molecule has two strands, and it gives the depiction of a ladder, so which is twisted on its axis. The sides of the ladder are made up of sugars and phosphates, and the rungs mean the horizontal support of a ladder. So the rungs are formed by bonded pairs of nitrogenous bases, namely adenine, guanine, cytosine, and thymine. So the sides of the two sides of the ladder are made up of 5 carbon sugars and phosphate rings, and the horizontal center part, the rungs of the ladder, are formed by the bonding of 4 nitrogenous bases, namely adenine, guanine, cytosine, and thymine. An A on one chain bonds to a T on the other, and similarly a C on one chain bonds to a G on the other. So that's how the basic chemical structure of a DNA, which constitutes genes, is made. As you can see in this diagram, this side and this side, these are two sides of a ladder. As you can see that these sides are made up of this 5 carbon sugar and a phosphate group, and this side also is made. This side of the ladder is also made up of these 5 carbon sugars and phosphate groups. But the middle horizontal rungs of the ladder are made up of bonded pairings between nitrogenous bases, namely cytosine, guanine, and thymine and adenine. So this is a basic structure of a DNA, or in broad sense DNA, which means which constitutes genes. Now we are going to talk about the physical structure of a gene. The physical structure of a gene consists of many elements, of which the actual protein coding sequences are often only a small part of it. Which means that these includes DNA regions that are not transcribed, as well as not translated regions of the RNA. This statement means that the structure of a gene consists of many elements, and those elements in the structure of a gene perform different functions, but most of the times along the length of the physical structure of a gene, not the entire part of a gene is encoded for proteins, and most of the part of a gene is not translated and not transcribed, and only a small portion of the length of a gene is actually translated and transcribed into functional proteins. Genes have an open reading frame flanked by promoter regions. Now, if we are talking about the physical structure of a gene, we first of all have to say that genes have an open reading frame. An open reading frame is the length of the gene where the enzymes read the codons and translate them into proteins. And these open reading frames of the gene are flanked by promoter regions. Promoter regions are present in the start of the open reading frames, which enhance and increase the rate and intensity of translation. Additionally, genes can have regulatory regions upstream or at the downstream ends of the ORF, and these regions of regulation alter the expression of a gene. Genes also have a terminator region where transcription stops. The ORF of a gene also has untranslated introns, which are removed before the exons are translated. So this means that the ORF, the open reading frame of a gene, also has some introns which are transcribed but never translated, and these introns are removed by splicing before the and only exons remain in the open reading frame, and only these exons are translated into functional proteins. As you can see, what I've explained earlier in this slide is very much easy to understand in this diagram. You can see that this is a depiction of physical structure of a gene. You can see that this is the regulation sequence or region which is present on upstream end of a gene and can alter the expression of gene after translation. This is the promoter sequence, which starts and enhances or increases the intensity and efficiency of translation. These are introns which are transcribed from DNA, but they are not translated into proteins, and these introns are removed. These introns are removed through splicing before translation, and only these exons remain in the open reading frame. This is the open reading frame from here to here, and only these exons remain, and these exons in the open reading frame are translated into functional proteins through translation. And as you can see, in this is the end part, and this is the termination region or terminal sequence. So this is a basic diagram of a gene structure, which includes regulation sequences, promoter sequences, open reading frame which has introns and exons, and the termination sequence. Now we're going to talk about the genetic code. The genetic code is basically a set of instructions inside a gene that tell the cell how to make a specific protein or a specific enzyme. The genetic code is the set of rules or regulations by which information encoded in genetic material, DNA or RNA, is translated into proteins, amino acid sequences, etc. by living cells. Adenine, cytosine, guanine, and thymine are the letters of the DNA code. They stand for the chemicals adenine, cytosine, and guanine and thymine. Each gene's code combines the four chemicals in various ways to spell out three-letter words that specify which amino acid is needed at every step in making a protein. So the genetic code basically is composed of four letters, which are A, C, G, and T, and these four letters are the nitrogenous bases: adenine, guanine, cytosine, and thymine. And these four letters, these bases, comprise the genetic code, and each gene's code combines these four basic letters into different forms and different combinations to create different amino acids. As you can see in this picture, these letters, this is the genetic code. It's like it's also called the language of life. GCA. These are the letters of the language of life, or the genetic code A, C, G, and when these letters of the code combine in the pairs of three, they constitute a codon. This is another diagram of the genetic code. As you can see, these are the letters of the code A, U, G, A, A, C, and when they are combined in the pairs of three, they constitute a single codon. You see, A, U, G, these are three letters are combined, they make one codon, and A, A, C combined make another codon. Now we will briefly discuss the history of the genetic code. The concept of codons or genetic code was first described by Francis Crick and his colleagues in 1961. During the same year, Marshall Nirenberg and Heinrich Matahi performed experiments that began deciphering the genetic code. Following this discovery, Nirenberg, Philip, Leder, and Gobin Khorana identified the rest of the code and fully described each three-letter codon and its corresponding amino acids. The structure of code letters. The portion of the genome that codes for a protein or an RNA is referred to as a gene. Those genes that code for proteins are composed of trinucleotide units called codons, as we've discussed earlier in this slide, that these are trinucleotide codon. Trinucleotide, or because a nucleotide has a sugar, a phosphate group, and a nitrogenous base, and each nitrogenous base serves as a single letter in the genetic code of life. So a trinucleotide or a nitrogenous base together constitute one codon. Each nucleotide subunit consists of a phosphate, as we've discussed this earlier. So this, as we have said, that a nucleotide, a single codon constitutes of three nitrogenous bases. We can also say that a single codon constitutes of three nucleotides, because one nitrogenous base is present in one nucleotide. So we can say that each codon is composed of three nucleotides or trinucleotide units. So to understand the structure of the code, or the letters of the genetic code, we must understand the structure of these nitrogenous bases which constitute the code. So there are four types of nitrogenous bases: adenine, guanine, cytosine, and thymine. There's also a fifth type, uracil, but that is present in the RNA and not in the DNA. So we must understand the structure of uh, these bases. The purine bases, adenine and guanine, are large as compared to the other ones, and they consist of two aromatic rings. But the pyrimidine bases, cytosine and thiamine, are smaller in comparison and consist of only one aromatic ring. You can see in this diagram that this is guanine and this is adenine. These are the two purine bases, G and A, and both of these consist of two aromatic rings combined together. This is guanine with two aromatic rings, this is adenine with two aromatic rings, and all the nitrogenous bases are called nitrogenous because they have amine groups, nitrogen and amine groups inside the rings. As you can see, they are the amine group, amine group, amine group, amine group, amine group. So they are called nitrogenous bases because they have amine groups and nitrogens inside their aromatic rings along with carbons. So these were the purine bases with two aromatic rings, and thiamine and cytosine are the pyrimidine bases which only have one aromatic ring, and also see one ring, and also have the amine group and nitrogen in their aromatic rings. So this is the difference between the four basic letters of the genetic code. The difference is based on their structure, the number of nitrogens, hydrogens, and carbons in their rings, and the number of rings in their structure. Now we are going to talk about codons. Because the genetic code is read and interpreted in the form of trinucleotide or nitrogenous based units, so we can't just simply understand the genetic code by understanding and studying one single letter or one single nitrogenous base only. We have to understand the codons, that how this genetic code works when they are in the form of three units and constitute a codon. So the codon is basically when three adjacent nucleotides constitute a unit known as the codon, which codes for an amino acid. For example, the sequence AUG is a codon that specifies the amino acid methionine. There are 64 possible codons, 3 of which do not code for amino acids but indicate the end of a protein, called stop codons. So in total there are 64 possible codons in the DNA, out of which 3 codons do not encode for any protein or enzyme, but they are simply perform the function of stopping or termination. So they are called stop codons. The remaining 61 codons specify the 20 amino acids that make up 20 essential amino acids that make up proteins, and out of these 61 remaining codons, one is called start codon. The AUG codon is called the start codon because it is universal, and it always starts. It's always the beginning of every messenger RNA, and that's why it's called a start codon. But along with being a start codon, it also encodes for an amino acid called methionine. So out of 64 total codons, one is the start codon, which also encodes for methionine, and 3 are stop codons, which do not encode for anything but simply perform the function of stopping and termination. There are only 2 amino acids out of 20 essential amino acids, methionine and tryptophan, which are encoded by just one codon. The rest of the 18 amino acids are coded by 3, 4, 5 or 6 codons. So out of 20 amino acids, only two amino acids, tryptophan and methionine, are encoded by a single codon, AUG and UGG respectively, and the remaining 18 are encoded by multiple codons. As you can see in this table, these are UCAG, UCAG. These are the letters of the genetic code, and when these letters are combined in trinucleotide form, they constitute a codon. And a single codon is responsible for an amino acid. As you can see that methionine, this red one, methionine, and this one, tryptophan. These methionine and tryptophan are the only ones which are encoded by only one codon. See, AUG, only one single codon is making methionine, and UGG, only one codon is making tryptophan. So these are the only two amino acids that are encoded by only single codon, and the rest of these, as you can see, are encoded by multiple codons. As you can see, leucine is encoded by 1, 2, 3, 4, 5, 6 different codon combinations. Valine is encoded by 1, 2, 3, 4 different codon combinations. Lysine is encoded by 1, 2, 2 different codon combinations. So through this table you can realize that you can easily understand everything about the genetic code and codon and combinations of codons and how amino acids are made, etc. This is just just a side note or an important tidbit, which is a very important thing to note, because only a while ago it was thought that the genetic code was universal. It was considered only in the past. It was considered that the genetic code was universal in all the species of all the organisms in the world. But recently it's been found that the genetic code diverges slightly in certain organisms and in certain mitochondria of some certain eukaryotes. So until the recent past, it was thought that the code of life was universal in all species of all organisms. But recently, the scientists have found that the genetic code diverges slightly in certain organisms and in certain mitochondria of some eukaryotes. So this is an important thing to remember. Now, what is the reading frame of the genetic code? The reading frame determines how the messenger RNA sequence is divided up into codons during translation. As we have described earlier, and the reading frame in this diagram we have described earlier, that the reading frame is the actual open reading, is the actual portion of the gene which is translated and transcribed. The portion that is actually translated and transcribed is called the reading frame or the open reading frame. And there are also some other like these regulation sequences, promoter sequences, the termination sequences are also present, but these are not translated or transcribed. So the only portion that is translated or transcribed, that is introns and exons, is called the reading frame. So the reading frame determines how the messenger RNA sequence is divided up into codons during translation. The start codon is the key signal, because the translation always starts with or begins with the start codon, and it keeps on going, and it continues in successive groups of 3. And the position of the start codon ensures that the messenger RNA is read in the correct frame. This statement means that the translation always starts when the start codon AUG is present on the open reading frame. When the start codon AUG comes up on the reading frame, the translation starts through different enzymes, and it keeps on continuing, and the translation keeps on going until the stop codon is reached. And in this way, a polypeptide chain or a protein is formed through translation. And the reading frame can be read in many different ways, and there is only one correct way to make a correct polypeptide chain, and that correct way of reading the codons is ensured by the presence of the start codon AUG. Codons in a messenger RNA are read during translation, beginning with the start codon and continuing until a stop codon is reached. Messenger RNA codons are read from 5 prime end to 3 prime end, and they specify the order of amino acids in a protein from N terminus to C terminus. This statement means, as I've discussed already, that they specify the order of amino acids in protein from N terminus to C terminus. The N terminus with an amino, this statement means that the translation is done starting from a start codon, and it ends at a stop codon from 5 prime end to 3 prime end. And the order of amino acids in a protein is from N terminus to C terminus. N terminus is the end of the DNA chain with an amino group exposed, and the C terminus is the end which has a carboxylic group exposed. In this diagram, you can easily understand the reading frame of the codons. As you can see that this is a DNA strand from 5 prime end to 3 prime end. This is the N terminus where an amino group is exposed, and this is a C terminus where a carboxyl group is exposed. As you can see that from this 5 prime end, the start codon is read, and from the start codon the translation process occurs, and from the start codon the translation starts, and it keeps on going until the stop codon is reached on this open reading frame. And at the stop codon of this reading frame, the translation ends, the translation stops towards 3 prime end, and that's and the start of the translation is from N terminus where an amine group is exposed, and it ends towards the C terminus where a carboxyl group is exposed. So this is a diagrammatic presentation of the reading frame and how the codons are read in the pairs of in pairs of 3 on an open reading frame during the process of translation from 5 prime end to 3 prime end, or from N terminus to C terminus, to create a polypeptide chain. So to sum this all up, everything we have discussed so far, if we are to sum all this discussion about the gene and the genetic code, we can in short simple statements we can say that the genetic code consists of the sequence of bases in DNA or RNA. Groups of 3 bases form codons. The codons are read in a sequence following the start codon until a stop codon is reached. The genetic code is universal. All known living organisms use the same genetic code. This shows that all organisms share a common evolutionary history. The genetic code is unambiguous, which means that each codon codes for just one amino acid, and there's no room for any doubt or uncertainty, and it's very certain, it's very unambiguous, because each codon codes for just one amino acid, or start or stop, so it's very unambiguous. Also, the genetic code is redundant, which means that most amino acids are encoded by more than one codon, as we've discussed earlier in that table of codons, that sometimes one codon encodes for only one amino acid, but sometimes 3 or 4 different combinations of codons, or sometimes 6 different combinations of codons encode for one amino acid. Also, to sum this all up, we can say that the genetic code is universal, unambiguous, and it's also redundant. That was it. Thank you very much. If you have any questions, you can please ask.